So welcome everyone to the 14th event of our Translational Neuroimaging Educational Program. In this program, we host a series of talks and presentations aiming to engage the neuroimaging and brain science community to enhance the translational application of computational neuroimaging techniques and promotion of human brain health. This is the 14th event now, which is really exciting. Just to give you an overview of the program itself, this educational program is nested within the lifespan working group of the Enigma Consortium. It is an inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. We hope to provide opportunities to expand skills, initiate collaborations, and facilitate networking in a welcoming learning environment. The program itself is directed by Dr. Ruyang Gi, who's on the call as well from the University of British Columbia, and myself. The program is also supported by our core advisory committee that you can see here. And there has been growing interest in the program worldwide, as you can see here. And the website where we have all the information on upcoming events is translational-neuro.org. You can also subscribe to our newsletter where you can receive updates of upcoming events, as well as news that we post on the website. All sessions that have taken place as part of the platform are uploaded to our YouTube channel as well, which you can access using the QR code here. Or you can also just search for translational-neuro on the YouTube website, and there you will see all the presentations and talks that have taken place on the platform so far. And this one will also be uploaded there as well. You can also follow us on Twitter or X, as well as Blue Sky for those interested, to, again, get updates on when we have upcoming events and any news that we post. You can follow us there as well. And additionally, we're always open to collaborations. If you have developed a tool that you would like to share with the neuroimaging community, please don't hesitate to reach out to us. We'd love to host you as part of the program as well. Okay, and moving to our presentation today, I have the pleasure of introducing Dr. Laura Hahn. She's an assistant professor at the Amsterdam University Medical Center. Her research is focused on the complex links between major depression and age-related biology. Using a wide range of neuroscientific methods, she aims to identify determinants of age-related biology and mental ill health, study their longitudinal dynamics, and evaluate prognostic potential and reversibility following treatment. We're very excited for you to present your talk today on brain age in major depressive disorder.
Yeah, thank you so much, Laila, and thank you so much for everyone for joining virtually today. Let me share my screen. Can everyone see the slides? Let me just pull up the laser pointer. Laser pointer, yeah. All right, yeah. So my name is Laura Hahn, and I'm an assistant professor at the University Medical Center in Amsterdam. And yeah, I want to talk to you about depression and mental disorder. So people with depression tend to age faster and live shorter. And a recent study has actually shown that mental disorders may reduce the life expectancy up to as much as 20 years. But I think the reasons for this are still, quite poorly understood. And I think it's very important that if we can identify what drives biological aging in individuals with depression, we may be able to prevent, delay, or halt abnormal aging trajectories. So a question that then kind of arises is, how do mind states influence biological aging and vice versa? And I think the answer here is not so straightforward. I think depression is a very complex mental health condition, and it interacts with equally complex biological patterns. And so it is then maybe not so surprising that their interactions are not easily or simply quantified either. So biological aging can be measured and operationalized in many different ways and can be derived from different functional levels or biological levels. But I think simply put, a biological aging indicator should at least predict chronological age or distinguish young from old, but also provide additional information about age-related risks that are there beyond chronological age. So to give a little bit more background and context to my research and myself, kind of want to zoom out for a moment and share how I got here. So I studied neuroscience in Amsterdam, so both my bachelor and master's degree. And I kind of started my first research internship over 10 years ago. And looking back at that time and then kind of fast forwarding to today, I think a lot has changed, and not just for me personally, but also professionally and in the field of neuroscience. And I think these changes have really shaped how we study biological aging in major depressive disorder. So back in 2013, I was really working on telomeres, so cellular markers of aging, and how they were related to brain function, and also to, for example, structural brain measures like hippocampal volume as a measure of brain aging. And I think these were really kind of small case control studies that had about 50 participants per group, and they were quite hypothesis driven. And then by 2016, when I started my PhD, I think things have changed quite a bit. So I think new technologies made it easier and cheaper to really screen kind of large scale biological data or different types of omics data. And we also had more access to computational power. And I think that led to a little bit more kind of data driven or hypothesis free approaches and different approaches, kind of similar how genetics kind of moved from single genes to the entire genome. And at that same time, I think the brain imaging field was going through a bit of a replication crisis, and scientists really realized we needed to have more robust methods, standardized data collection, harmonized pipelines, large sample sizes. And this is where I think data sharing really became crucial, not to increase or not only to increase statistical power, but also because I think brain scans are quite expensive, and then sharing really makes the most of every data set. So I had a front row seat to the rise of Enigma, which you are probably all very familiar with. And I've been involved in many of their collaborative projects since. So by the time of my PhD and then later also my postdoc, I think three big things had changed. So we had big data, much more and diverse biological data, smarter analyses. So we needed more intelligent tools to make sense of it all, and a scientific infrastructure and systems that had to store and process all these data at scale. So I think these were really exciting times for me to be doing research, and I'm looking forward to sharing with you some of the findings of my own work. And of course, towards the end of the presentation, I'll also touch upon what we still don't know and where the field might be heading next. So the findings that I'll share today are based on what's called an age prediction modeling framework. And although you're probably already familiar with this framework, I still want to briefly explain it. So in simple terms, we kind of use machine learning, mostly supervised learning, to estimate a person's brain age or biological age from their brain data. And the idea is to teach a model how to predict someone's age based on patterns from the brain scan. So you can use different types of data for this, and I've mostly used DNA methylation or structural MRI scans. But today I'll focus mostly on the predictions made from structural MRI data. And so if you look in A, we kind of first use MRI scans from a group of healthy individuals whose chronological age we know. And these scans are then used to train a regression model, which learns the relationship between brain structure and age. And then if you look in B, we check how accurate our model is. by using tenfold cross-validation, so dividing the data sets into 10 parts and then training the model on 9 parts of the data and testing it on the remaining 1. And we repeat this, so every part is tested once. And then we compare the predicted ages to the real ages to assess how well the model is performing. And then once we're kind of confident in the model's accuracy, we can use it to predict biological age or brain age in new individuals, but also, for example, in people with depression. And these predictions are independent of their actual age and gives us an estimate of what their brain looks like biologically and whether it's older or younger than expected for their age. So my passport says that I'm 35. It still says 34, and I'm actually 35. But if I showed this algorithm my brain scan, it would probably say that my brain looks, of course, much younger. And the gap between your actual age and your predicted brain age is the brain age gap. It's a simple number, but a powerful one. And the difference score can be statistically linked to other traits. And it has been shown to predict important outcomes like mortality better than chronological age. And so, in other words, the brain age gap kind of gives us insights beyond what the calendar says about how someone is aging biologically. So over the past decade, the field of biological aging research has really grown rapidly. We've seen major advances in identifying markers of aging, and including those used in mental health conditions like depression. But I think when coming to translate these findings into clinical practice, progress has still been quite limited. I think there are a few key reasons for that. So one, there are small and some inconsistent studies, relatively small sample sizes that are using different methods, leading to heterogeneous findings. But also group level findings, so not the individual level predictions. So most progress so far has been at the group level. And we might find that on average, people with depression have older biological brains, but it doesn't help us predict aging in the individual patients, which is something that clinicians would really need. And also there's limited evidence still for clinical outcomes. So not many studies have really tested whether brain age gap can actually predict meaningful clinical outcomes, things like treatment response, relapse, or long-term health risks. So while the science is really advancing, I think we're still figuring out how to make use of it in real-world clinical settings. And during my PhD, I looked at several different biological age gaps across different biological systems, mainly using large-scale cross-sectional studies. And a key data set that I worked with was the Netherlands study of depression and anxiety, which includes nearly 3,000 participants. And about a third of those have been diagnosed with depression or anxiety. For brain-specific findings, I mostly work with imaging data from the Enigma MDD consortium, but I also worked with, for example, post-mortem brain tissue from brain banks to get additional insights on the cellular level. And I think what I want to draw your attention to here is that across the different biological systems and using various methods, we do find a consistent pattern. So depression is associated with an older biological age. But I think you can see here that while the effect is quite robust, it's also quite subtle. So even a small shift in biological age can reflect underlying stress in the body and the brain. I think it still raises an important question. So what kind of factors really contribute to biological aging, and how can we better understand their impact, especially in people with depression? So in the rest of today's presentation, I'll take a closer look at the findings from brain age and depression. And I've been very fortunate to collaborate with a really incredible group of researchers to develop a brain age prediction model, working closely with the Enigma MDD consortium. And thanks to its global network, we had access to data from 19 different cohorts around the world, giving us nearly 7,000 brain scans that covered almost the entire adult lifespan. And this kind of large-scale diverse data set is what makes it possible to build robust and generalizable models. And it really reflects the power of, I think, international collaboration in neuroscience. So to give you a better, can I maybe this? Yes. So to give you a better sense of the data behind our brain age model, this is a short video clip created by Jan-Mark Deeuwe for the Enigma Plasticity Working Group. And it kind of really illustrates how brain structures change across the lifespan. So some regions grow and others shrink, and some stay relatively stable over time. So from childhood through adolescence and young adulthood, we see growth in white matter, and while gray matter, especially cortical thickness, declines. And as people enter older ages, we typically observe further reductions in cortical thickness and hippocampal volume, while the ventricles tend to enlarge. And what's important to take from this is that these brain changes follow reasonably consistent patterns across individuals. And then we can really use those patterns to statistically capture how the brain structure normally changes with age, and then use these predictive algorithms to estimate someone's chronological age on their brain scan. And that's kind of the core idea behind this approach. So to build our brain age model, we used a rich regression, and it's a type of machine learning algorithm that works well with dealing with many potentially correlated features. And the model takes as inputs cortical measures, parcellations based on the Desikan-Kiliani atlas, and subcortical volumes from the ASEC atlas extracted using the FreeSurfer software package. And this gives us a standardized set of brain features that we can use. And I also want to take this moment to introduce the two people who played a key role in kind of taking this model beyond the research lab and implementing it into a web-based tool, making it more accessible and easier for other researchers to also use. So using this model, we found that patients with depression indeed had older brain age estimates. But as you again can see, the effect is really subtle with a lot of overlap between groups. Although we've replicated this as well in cohorts that joined our consortium later than the initial development of the model. But I think it's important to mention that if we have someone's brain age gap, we really cannot reliably tell whether it's a patient or a control. So it's not a suitable marker for diagnosing depression. But since then, I think we have fairly reliable and robust evidence that indeed the brain age gap is higher in individuals with depression in specific, but also mood and psychotic disorders more generally. And when we applied this model to different data sets around the world, we find that the model generalizes reasonably well to independent cohorts, in which I think it really has demonstrated clinical validity by being able to pick up on associations with psychopathology. So I really want to emphasize that a higher brain age gap is not specific to depression. It's more of a general transdiagnostic indicator. I think it says something more about compromised brain health, and it's related to other psychiatric disorders as well, such as schizophrenia, bipolar disorder, specific phobia, but also other medical emergency that impact the brain, such as stroke. And taken together, I think the magnitude of the brain age gap kind of follows a pattern that is consistent with other structural brain findings, and namely that we see kind of the largest effects in schizophrenia, with smaller effects in, for example, bipolar depression and anxiety. So we know that people with depression and anxiety tend to appear biologically older than their age-matched peers, but we don't fully understand why or what are the factors that cause these deviations from the normal aging process. And so one of our key goals is to uncover these drivers, and that means that we're looking at things like pre-existing vulnerabilities like genetic risk, but also modifiable lifestyle factors, such as, for example, BMI and smoking. And kind of by separating these influences, we hope to better understand what contributes to a higher biological age in depression. But whether an older appearing brain is a cause or a consequence of depression, I think is still an open question. We know that many different factors contribute to it across the lifespan, and some studies even suggest that this gap is more a reflection of a vulnerability to early life factors rather than something that predicts how the brain changes over time. In another one of our collaborative studies, using data from almost 4,000 participants in the Enigma MDD working group, we found that a higher brain age gap was associated with an increased genetic risk for depression and inflammation, as indicated by CRP. And we also found that early life adversity played a role. But I think the most interesting finding here is that the most robust and unique associations were with modifiable lifestyle factors such as BMI and smoking. And this highlights, I think, the potential to target lifestyle interventions to support brain health in depression. And this approach, may still be quite underutilized in clinical practice. I do want to mention that is still based on cross-sectional evidence. So I think the next step is to look at this in more trial perspectives. I think we still have much work to do in evaluating the clinical relevance of brain age and biological age for treatment. But so far, I think there are some suggestions that there are associations with modifiable lifestyle factors. And I think this provides some more neurobiological basis for incorporating lifestyle interventions into depression treatments. Again, so far cross-sectional evidence, but another research question that I'm interested in is the clinical utility of brain age. And so it's unclear whether, for example, a higher brain age at the start of treatment can predict response or clinical outcomes. And it's hypothesized that an abnormal brain age will be linked to a poorer treatment response or lower remission rates and worse outcomes. And if this is the case, then this would allow clinicians to use it as a predictive tool for treatment success. But as you can see here, there are so far only a handful of studies that have looked at the predictive value of baseline brain age to predict treatment outcomes in depression using different types of brain age models. So leading to a bit of inconsistent evidence here, where some studies do not find an association, other studies find that a higher gap leads to less symptom reduction. And then this is pretty much work in progress, but here we find that a higher brain age gap predicts greater response rate, but also on the other hand, that it predicts less symptom reduction. Also in a variety of different treatment designs, but most of them focusing on antidepressants. Another key question in this field, I think, is whether the brain age gap is dynamic. So in other words, can it change over time, and can it potentially be used to track treatment outcomes? And there's a lot of hope and interest in this idea, especially because it could make brain age a valuable marker in clinical settings. And I want to highlight two studies that suggest that there might be dynamic potential. And this first is from Lei and colleagues back in 2018, where they showed that healthy individuals who took ibuprofen for 8 weeks had a reduction in their brain age gap compared to those on placebo. And the authors here suggest that this might be a reflection of the anti-inflammatory effects of ibuprofen acting on the brain. And then more recently, a study was published that shows that individuals with relapsing remitting multiple sclerosis were able to reduce their brain age gap after 6 months of treatment with bexarotene compared to a placebo group. And here the proposed mechanism is remyelination, particularly in the cortical gray matter lesions. But to date, I think the evidence is still quite limited. Now, when looking at trials specifically for psychiatric disorders, the current literature is also quite limited. And I want to again mention two different studies. So the first one was conducted in individuals with first episode schizophrenia, where the rate of change of the brain age gap did not differ or worsen between responders and non-responders over a 12-week treatment with risperidone. And the second study I want to highlight is one of my own, where we found that baseline brain age gap did not predict any symptom changes over 12 months in first episode mania youth treated with lithium or quetiapine. And the brain age gap also remained stable over time and was unaffected by treatments. So I think despite the growing research interest in brain age as a framework and kind of many opportunities for secondary analyses using existing trials that have repeated MRI available, only a handful of studies have been published on this so far. And yeah, I'm not sure why this is, but my sense could be that either people find it challenging to access the different types of models or select the right model for their data set, or potentially maybe the field is facing a publication bias where maybe null findings remain unpublished. Now, given that much of the literature does seem to point to underlying inflammatory pathways and metabolic dysregulations as potential mechanisms underlying the brain age gap, we were very eager to study whether a lifestyle-based intervention using running therapy would be able to point to dynamic changes. And so we used participant data from the MOTAR study, so mood treatment with depressions or mood treatment with antidepressants or running therapy, and which is conducted between 2012 and 2019. So very briefly, the trial investigated the effects of a 16-week intervention with either antidepressants or running therapy in patients diagnosed with depression and or anxiety disorders. The study had a partially randomized preference design, so participants without a strong treatment preference were randomly assigned to either intervention. And so the primary analysis of this trial showed that running therapy and antidepressant medication had similar effects on mental health, so similar remission and response rates. And this really demonstrated that running therapy is a valuable treatment strategy in mental health care. However, the groups also differed significantly on various changes in physical health. So for example, weight, waist and circumference, but also it's really not only an improvement of several of these physical health markers in the running group, but also really a worsening in the antidepressant group. So for example, inflammation, heart rate variability, and weight gain was observed in people using antidepressants. And I think because we previously observed these associations between more metabolic, somatic, inflammatory markers and biological age indicators, we were hoping to see whether running therapy might potentially have differential effects on the brain age gap as well. So now when in deciding what kind of model to use to obtain predictions, our initial intuition was to go with a model that has more accuracy or a better performance in predicting age. So we used a deep learning model that has shown excellent performance accuracy and has been trained on the largest and most diverse assembled training data to date. But there's also some evidence showing that there is a divergence between model performance and clinical utility. So if you look at the left panel, our work within Enigma MDD showed that in these 13 participating cohorts, there was actually no relationship between the model performance and the effect size of the between group differences, so the case control differences, but also not between the estimated brain age gap between the groups. And then on the right-hand side, you can see that a systematic review published in Patterns similarly showed that model accuracy was also not related to model utility, and model utility here was then defined as the absolute correlation between the brain age gap and the studied phenotype in that study. Then Basham and colleagues also provided evidence that showed that moderately fitting brain age models obtain significantly higher differentiation compared to tightly fitting models. And because of this, we also opted to compare two different brain age models for the current study. So I won't go into too much detail, but to put the upcoming findings into context, I think we must appreciate the features that are used to train the different models. So the first, again, is the one that we developed. It's a very simple model based on 77 free surface-derived measures of thickness, surface area, and subcortical volumes. And the second is a convolutional neural network trained on voxel-wise patterns without explicit region-based feature extraction. So really using more low, mid, and high-level features of the brain. And here you can see that as expected, the deep learning model really outperformed the simple model on the left-hand side, although the simple model also still showed reasonably good performance accuracy, including test-retest reliability. And when comparing cases and controls, the PyMant model on the right-hand side showed a small but non-significant case control difference with an effect size of 0.17. That's a Cohen's d. And in contrast, the Enigma photon model estimated a larger mean difference of the brain age gap between groups, but also had a moderate effect size of 0.44. Now, when assessing whether the brain age gap could be dynamic, we conducted a linear mixed model with the brain age gap as the outcome, and then time, treatment, and a time by treatment interaction as predictors, but at the same time also correcting for age, sex, and Euler number. So Euler number as an indicator of image quality. And what I want to draw your attention to is that while the regression coefficients of the predictors of interest are quite consistent in terms of the direction of effects, the Enigma photon model yielded stronger coefficients. And in terms of significance, there was only a trending effect of the time by treatment interaction in the Enigma photon model and a non-significant interaction in the PyMant model. But when correlating the brain age gaps from both models while also partialing out age, we found that there was a relatively low correlation between the two different brain age prediction models, suggesting that the gaps may capture complementary aspects of the brain age variance. And I think one advantage of this is that I could therefore integrate them into a combined score. And by integrating both scores, we found stronger effect sizes that or coefficients that better captures or outperforms the treatment impact than individual scores. And as you can see from this plot, where we have the brain age gap on the y-axis that is residualized for age, sex, and Euler number, and then the 2 time points before and after treatment on the x-axis, you can kind of see that, yeah, the spaghettis do show that there is quite some individual difference there in how dynamic the brain age gap is. But so yeah, several things that I want to draw your attention to. So looking across groups, the brain age gap does seem to show dynamic change over time. But I think what you can see here is that while individuals in the antidepressant group show an increase in the brain age gap following this 16-week treatment, this effect was not so apparent in the running therapy group. So if we're talking about dynamic change, I think it seems it's not so much a reversibility or normalization of brain age following running therapy, but more of a worsening of the brain age gap in the antidepressant group. And I think that that's kind of consistent what we found in general in the primary analysis of this study. Now, this is very preliminary still, so this finding still really requires some follow-up. But my intuition here is that this might be driven by the psychotropic weight gain and changes in inflammation, as we've seen for CRP. So some of the conclusions, baseline findings, we confirm, and that's consistent with literature, that we see a higher brain age gap in people with depression and anxiety disorders. And in terms of clinical utility, running therapy may help to halt further progression of poor age-related brain health. And antidepressants, while improving mental health, may have unintended negative effects on at least age-related brain health, potentially due to the adverse effects on physical health. Some methodological insights, so models with moderate fits may better differentiate clinical groups. And in using the Enigma photon model, the advice here is, the recommendation is to include Euler number as a covariate to control for image quality. And both brain age models showed consistent coefficients, but different magnitude. But a way forward potentially in looking at different types of models at the same time is to combine different brain age models, as it may enhance sensitivity, as shown that when we integrate multiple brain age gaps, it shows a stronger detection of the treatment effects. So which model should be used and what are kind of the challenges that we're facing? I think the first challenge is deciding what kind of model to use, and it's important to realize that a model with a higher prediction accuracy doesn't always mean that it's more clinically useful. And we see here that models with only a moderate fit actually show a stronger differentiation between clinical groups. But I think there are a lot of pragmatic and logistic factors here as well that drive the choice of models. So not only what type of data is available or what type of data can you share with other groups and collate and aggregate together, but also computational research or resources of your lab, or maybe access to collaborators that use a specific model or have specific expertise. And I think this really also shapes kind of what is practically feasible in many studies. And another concern is selective reporting. I think there's a kind of a risk here of testing multiple models, but then only reporting or publishing significant findings. And I think that can really distort the evidence base as well. But also, I think, even selecting a model is quite complicated. The literature is quite fragmented. We use brain age gap for pretty much any type of modeling that predicts age, but also depending on, sometimes it's multimodal, sometimes it's gray matter features, white matter features, resting state or functional MRI. So I think, the literature is quite fragmented, and it makes it difficult to compare the different studies with each other. Also how to evaluate and compare them to each other. So I think this really highlights the need for more consistent and standardized language. And on top of that, I think gathering the code for different models and running systematic comparisons really takes substantial time and coordination. And I think here we can really learn from the field of epigenetics, where you have more centralized, more standardized toolkits that exists for calculating multiple different clocks at the same time. I think, or I do acknowledge that with imaging data, this might be way more complex, but I think there's clearly a need for a similar kind of centralized repository for brain age models. And then finally, I think transparency is essential. I think there's growing enthusiasm around brain age research. So null findings may be underreported, and I think this makes replication efforts and careful interpretation of results more critical than ever. I want to acknowledge all the people that I work with and my collaborators, the funders, and of course all the participants and contributors to the studies. And I now have a short live demo of how to use the Enigma brain age model and the web-based tool for that. So it's always a little bit exciting to do a live demo. So let me see If I can pull up the right screen for that. Yes, can you see this website?
Yes.
Yes. Okay, so the website is photon-ai.com/enigma _brain age. And what you can do here, so if you run recon all on your data and you have your free surfer output, the way to go about it is to yeah. This is kind of standard Enigma protocol of how to extract the cortical features and how to QC the cortical features as well, how to organize them. And you may, if you are familiar with the Enigma consortium or you have contributed data to there, these should be quite familiar to you. And the same goes for the subcortical features, where you can also go to this link where it will also go to the instructions how to extract subcortical volume features. So what I've done is taken the scripts to extract the features from the free surfer recon all outputs. So if you can see on this side, this is my recon all free surfer output. And if I kind of go to that folder, so this is the output folder, the same as you see here. And what I do then is extract the the cortical features. I've timed this. It should take about 30 seconds. And what it does, it will create cortical measures, free surfer derived ROI, so thickness and surface area measures. Give it a couple of seconds there. Important, of course, before going through these steps is to QC your data as well. And Enigma has standard QC protocols that you can follow to do this. So that creates these two files. Then I can also extract the subcortical volumes, which are here. And so what you can do then is just take these output files and compress them into a compressed folder, a zip file, go to the website and input your name, lab. Oh yeah, it's also important to mention that we trained separate models for females and males. And so that's something to take care of in analyzing your data. So when you upload your zip file, you can get predictions. So these first ones are the females. And I'll now apply the ridge regression coefficients for the male model. That gives this output. And what you can then do, and I'm happy to share these kind of scripts, but they're very standard, is to just kind of have your standard covariate file. So in this case, I'm having subject ID, diagnosis, sex, and age. And then because the file that is output from the website does not include any subject IDs, you will need to take the subject ID list from the input file that you used as a cortical measure, for example. Then you can load the brain age predictions. So for females, males, merge the subject IDs, and then merge them to your data set. So you would have to match on what is a male or what is a female subject. And then you can just go about looking at your generalization, how well is the model fit in your data set, and then plot, for example, what it looks like in terms of model fit. And as you can see here, we do have this the age bias where we see that younger persons are estimated to be older and older persons are estimated to be younger. So you would need to correct for the age bias in some way as well. But I think, yeah, it's relatively easy to get your predictions in this way and then just go about your subsequent statistical analyses in what your research questions and hypotheses are. So I think I'll stop here, and I'm very happy to enter in a discussion and have any questions if you have, answer your questions if you have them.